I'm Gerran Collins.
And I'm Kayla Allen, and this is Genetically Modified Crops and Animals.
What is a genetically modified crop and animal? A genetically modified crop or animal would be any organism that has been genetically modified with genetic engineering techniques. You may recognize some of the images on the left from your local grocery store. These are common genetically modified food sources. Some of the top genetically modified crops are cotton, squash, corn, potatoes, soybean, canola, apple, and papaya. Wheat, rice, and tomatoes are approved for the market, although they are not used. The top genetically modified animals are pigs, cows, chickens, salmon, and even mosquitoes. Mosquitoes were modified to help stop the spread of the Zika virus. When did this begin and why? The first genetically modified crop put on the market was the Flavor Saver in 1994. This was because tomatoes had a very short shelf life, so it was hard to keep them in good conditions while being transported. The Flavor Saver was created to slow the ripening of the tomato. The Flavor Saver had a lower level of polygalactorinase, that is also known as the PG gene. This made the fruit cell walls degrade slower because of the lower levels of PG, although actually the first genetically modified crop was tobacco. Many people do not know this since it was not made for nutrition like the Flavor Saver. That is why many people say that the Flavor Saver was actually the first. The tobacco plant was genetically modified in 1983. The reasoning was to create a genetic gene that joined with the antibiotic resistant gene to the T1 plasmid in agrobacterium. This allowed the tobacco to be herbicide tolerant. The first genetically modified animal was a mouse in 1974. This was done to show the metabolic rate. They were genetically modified to have strength from increasing the muscle growth. This was done by overexpressing the insulin growth-like factor in different muscle fibers. The image shows the knockout and knock-in, referring to the Cre recombinase gene. Where is more research needed? I think that there could be more research in the ethics and how GMOs are presented. Many people, when they hear GMO, think that it's a bad thing. I was even one of those people. It is because we do not have the information, so we are left to believe what others are saying around us. We do not know that most of the things that we eat, all the way from apples to chicken, have been genetically modified in some way. It is also not easy to find information that you can give to someone who's not a science student that makes sense. If there was a less complicated information, more people would understand it. Another issue is the ethics. People believe that you are hurting the organism and GMOs are ethically cruel. More research would help stop the rumors of animals being hurt or tortured, as well as the thought that you are eating a crop that's filled with chemicals. This would help show that GMOs are not something that you should fear. GMOs will not hurt you or the organism. Another interesting thing is that we are actually GMOs too. This is because whenever you get a virus, they insert their DNA, and this alters your genome. I think that putting that in people's mind would be another way to stop the fear of GMOs. What is the price range for these procedures? There is not a significantly cost difference between farmers producing GMO versus non-GMOs. The cost difference for most GMO items is in between the cents. This makes it not hard to produce a GMO, especially if you are a farmer that's switching from non-GMO to GMOs. Many farmers actually make more money growing GMO crops and animals since they can sell more than they did before. A good example would be apples. GMO apples are bigger and they look more appetizing. If a farmer is getting paid by the pound of apples, they would make more since the GMO apples are bigger, as well as more people would buy the GMO apple because of how it looks. If the produce is herbicide tolerant like Roundup Ready crops, the farmer would spend less money and time caring for the crops. Although there is a downside. It does cost a lot to actually create a GMO. The discovery, development, and authorization are all major parts that take time and money. Since there is a lot of technology going into this, as well as labs, manpower, and other components, this makes a high price. Normally, the average is around 136 million. On the bright side, though, if you are able to make it through the process, you will easily make your money back and more. GMO 1 through GMO 4. GMO 1 is selective breeding, so this is selectively crossing an individual to get the desired trait. This will also allow you to see what the future generation will be. GMO 2 is hybridization. This is better done with plants, so you would do this with a crop. Crossing two different species that have closely relate. An example would be a mule. GMO 3 is mutagenous. This is chemical or radiation. This is big changes by altering the genome. The one we use the most would be a GMO 4. This is modern genetic engineering. The advantages are it's faster, very specific, and it happens in the natural world via horizontal gene transfer. This is when a bacteria or virus shares or exchanges their DNA.
The steps. Today, GMO 4 is the most widely made GMO because the process to make them is relatively easy, at least compared to the other GMOs it is. Because of this, we will be talking about the process of making GMO 4. This process can be condensed down to 5 basic steps. Those steps are isolating your gene of interest, constructing the recombinant DNA molecule, putting the recombinant DNA into a bacterial cell, putting the recombinant DNA into a plant or animal cell, and testing transgenetics. That means testing to see if the gene was successfully taken by the genome. So let's break down these steps a little more. Step 1 is isolating your gene of interest. There are 2 ways to accomplish this step. Polymerase chain reaction, or PCR, is one of the ways and is not really the best way. The second and most common way is by using a restriction enzyme. Using these enzymes is considered the best way to isolate the gene. These enzymes recognize and cut specific sites on DNA fragments. The most commonly used of these enzymes is EcoR1. EcoR1 is a naturally occurring enzyme in bacterial cells. All of this cut up DNA is not just laying on the table ready to be picked up through. It is in a tube all jumbled together. In order to separate the DNA we want from what we don't want, we use gel electrophoresis. This will separate the DNA based on molecule size, and because we know the size of the DNA molecule we want, we can harvest the specific one from the gel. Step 2 is constructing the recombinant DNA molecule. Recombinant DNA is DNA that has been artificially formed. There are 2 things needed in order to form this DNA: the gene of interest and a vector. The vector is also called a plasmid because it is a plasmid from a bacterial cell. In order to get the plasmid to take up our gene of interest, we have to cut circular DNA. We cut it with the EcoR1 enzyme, making a linear piece of DNA with sticky ends. Our DNA molecule we cut earlier will also have sticky ends. The sticky ends from the plasmid and our DNA molecule will be complementary to each other and bind together. A special enzyme called ligase will be used to repair phosphodiester bonds in the DNA backbone. But this is not all plasmids have for us. They also carry promoters and selectable markers. Promoters will tell when to turn our gene on and off, and selectable markers are usually antibiotic-resistant genes that will help us determine if the recombinant DNA was taken up later in the process. Step 3. Inserting recombinant DNA into a bacterial cell. The bacterial cell that is used is non-pathogenic E. coli. In order to get the recombinant DNA into the bacterial cell, we use heat shock. This is where the recombinant DNA and bacterial cells are placed together in a tube and then placed on ice to cool for about 10 to 15 minutes, and then taken off of the ice and immediately put into warm water. The shift in heat causes the disruption of the bacterial cell's plasma membrane, allowing the recombinant DNA to float in, but not all cells will take up the DNA. The cells can be tested for the DNA by placing them on an agar plate with food for the bacteria and whatever the selectable marker is supposed to fight against. The ones that live have taken up the DNA, and the ones that died did not. To replicate the bacterial cells, put them in a liquid medium. They will also replicate the recombinant DNA. Step 4 is inserting recombinant DNA into a plant or animal cell. For plant cells, there are 2 ways to accomplish this step. Biolistics or Agrobacterium tumefaciens. Biolistics is where scientists coat plastic or gold beads in the recombinant DNA and literally shoot it into a plant using a gene gun. Agrobacterium is a common soil bacteria and is a natural genetic engineer. Normally, Agrobacterium inserts genes that are lethal to plants, but scientists will take out these lethal genes and replace them with the recombinant DNA made in the previous step. There are several ways to insert the recombinant DNA into animal cells. Microinjection is one of the simplest ways. This process uses a microscopically fine glass micropipette to inject the recombinant DNA directly into the nucleus of the animal cell. But not all cells are big enough for this technique. Electroporation is a technique that makes artificial pores appear in the membrane of the host cell. This allows the recombinant DNA to enter the cell. Liposuction uses a lipid complex to deliver DNA to cells. Sperm-mediated transfection uses sperm's ability to spontaneously bind to and internalize exogenous DNA and transport it into an oocyte. Although there are plenty of ways to make genetically modified animals, they are not used often in GMO research. This is because they are not as readily admittable to genetic transformation as most plants, and because it is seen as unethical, even though it happens in nature all the time through horizontal gene transfer. Step 5. Confirm transgenics. The reason we are making GMOs in the first place is to make more, better, and longer lasting products. So if the product isn't doing what we need it to, then what was the whole point in this entire process? The way we confirm this is simple. For plants, you can either do PCR or simply taste the fruit. That's right. You don't have to go through lots of experiments to confirm transgenics. Most of the plants modified are to make bigger, tastier, and longer lasting fruit. Take the apples at the bottom of the screen. The one on the right was modified so that it would not turn brown as quickly as the apple on the left. This is supposed to help cut back on food waste. Even though it's brown doesn't mean it's not edible. Most animals are modified to be bigger, meatier, and produce more offspring. The fish at the bottom of the screen are different in size. The top one is a GMO, and the bottom is not. That means the consumer is getting more meat for their money. What are the benefits to genetically modifying crops and animals? GMOs have higher yields, longer shelf life, and are resistant to disease and pests. Because of the longer shelf life there is great potential to cutting down how much food is wasted each year. Many stores and people will throw out bruised or slightly changed fruits and veggies because they look ugly. With higher yields and not having to spend money on herbicides or antibiotics, products are cheaper for the consumer to buy. Not to mention they will be healthier as well. The environment also benefits from this because there is no runoff from the herbicides. What are the cons to modifying crops and animals? Although there are many pros to GMOs, there are also cons. Organisms are changed in a way that is not naturally occurring. Take the apple from before. That fruit is not going to decide to slow down its decaying process just because someone didn't buy it in a store. There is a raised risk for allergic reaction, although this is a very low risk. This could be something like taking the genes from a peanut and adding them to a tomato, and then all of a sudden you have hundreds of people in the hospital. GMOs can change many factors about an organism and its environment. Take tobacco, for instance. It's a GMO. When its leaves fall off and start to degrade, the nicotine from these leaves seeps into the ground and kills all the surrounding plants. But if scientists are careful and know what the genes they are using do and how they react to environments they are being put in, then these are all very low-risk cons.
How the benefits outweigh the cons. Is it really worth it? We are very dependent on genetic engineering since it produces more amounts of food. An example would be the larger sizes in chicken. That gives you more meat to eat. The world does not produce enough food to sustain everyone naturally. The risks with cons are relatively low when compared to the benefits. The con of changing the natural way and other factors of an organism is outweighed by creating a better form of that organism. These organisms are improved instead of being completely altered. There can be the con of allergies, but it rarely does occur. And I think that the biggest risk and con would be the changes to the environment because of the GMO. A GMO can cause disruptions to an environment and other organisms that are residing there. You have to make sure that you watch what genes are going into the GMO so that you do not disrupt the environment too much. It is worth it because scientists are looking at the bigger picture of helping make better things. The goal is not to change everything, but instead to improve them. This is so that we can benefit. I think that this is important because it's helping make things easier and more accessible. Also, the changes being made are relatively small. They want to make subtle changes that benefit us instead of things that would end up hurting us in the end. Ethics. Ethics focuses on caring for others and acting in a way that maximizes the happiness for everyone involved. I think that that is important because we are caring for these plants and animals like we should, and not treating them in a bad way. Kant explains in his first formulation that we should act in a way that everyone can. This ties in with GMOs by making sure all manufacturers are acting in the same good and safe way. He wants everyone to follow rules so everyone's on the same page and no issues arise. He explains in his second formulation to treat all beings as an end and never as a mean. So that means that you cannot use organisms simply to get what you want. Instead, you have to treat them right, since they are alive just like you. He also implies that we should not lie. That means GMO products should be labeled so we are not lying to the consumers. GMO labeling will not solve all the problems associated with GMOs. However, it will allow us to make a free and informed choice about whether or not to consume a GMO. It also allows scientists in all fields to better document the impact of GMOs on the environment, health, and the economy.